we're getting into linked genes here. So here we have a typical dihybrid cross. Here to draw a Punnett square. So what we're not showing is the parental and the F1. This is the cross that you do if you did the F1 cross, right? So you get your 9, 3 to 3 to 1, where these guys are the recombinants, right? These are the parentals. They look like the parents. So we expect a 9 to 3 to 1, right? In 1905, Bateson and Punnett, the person who came up with these Punnett squares. Actually, I think two people did. They used to be called Punnett and something or other squares, and that was too hard to say, so people just call it Punnett squares. Hey! Good, you? Welcome to class. All right. So, here we go. We've got an organism that has purple flowers and long pollen. And we cross that with an organism that has red flowers and round pollen. And the progeny of purple flowers and long pollen. What's dominant? Purple or red? Purple. What's dominant? Round or long?
Long.
Right, okay. So great, we know it's dominant recessive. It didn't look like there's any kind of blending that occurs. Let's run the actual cross and get the data. So we would expect a 9 to 3 to 3 to 1 ratio, right? 9 look like this. 1 are going to look like this. This is recessive and recessive, right, for both characteristics. So 9, 3 recombinants, and 1 of this. We would expect that, and when they ran this experiment, they found a 15.5 to 1 to 1.5 to 4.5 ratio. Not even close to 9 to 3 to 3 to 1. There were other experiments and there were other people running experiments at the same time. They were finding traits that didn't follow the 9 to 3 to 3 to 1 ratio. And so people were trying to argue that this isn't right, there's something else going on. These traits of pollen type and flower color do not follow the law of what? Segregation or independent assortment. Dihybrid cross, independent or assortment. 2 words, right? Right. So this appears to reject that. Another researcher, I think his name is Thomas Morgan, 1911. Thomas is the one who found the white mutation, the white eye mutation in Drosophila. Morgan was really important. Morgan had a larger number of graduate students go through his lab. And all of those graduate students went on to become geneticists, and they all studied Drosophila. And many of them found very important things on their own. And so Morgan was kind of like this there's populations on Earth where, like there are, I've heard, I don't know if it's true, like people over in some countries, they'll move in, like Islam nation will move in, and the people will try to outbreed the native population there, so they become the majority population. Does that make sense? That's what Thomas Morgan did. He had so many people go through his lab and going out and getting positions as geneticists studying Drosophila that's why we study Drosophila today. Because he was so successful in getting people in and out and placed and established to solid careers. Isn't that funny? That's how it works. So, he studied 3 traits, and these guys appear to violate this law of independent assortment. Eye color, body color, and wing type. So their red eyes are wild type, but white eyes are not wild type. They're the recessive mutant. And then there are flies that have large, normal-sized wings, and then there are wings that are called miniature, smaller wings. And then there is body color, gray or yellow, and wild type is gray, and yellow is the mutant. So he was crossing these traits, and he found that he was not getting the 9 to 3 to 3 to 1 ratios, or the expected ratios. And he was actually also following this in sex-linked crosses also. So here's the actual experiment that he ran, and I won't go into all the details, but he had a male with certain characteristics and a female with certain characteristics. He found that these were all in the X chromosome, and they were not assorting to, the way he set this experiment up, he would expect equal numbers of each possible phenotype, and he did not get that. I won't go through the details of this. So what could possibly explain this? All three genes are located on the same chromosome. They're all located on the X chromosome. So since the X chromosome is inherited as one whole unit, when you inherit one thing, when you inherit an X chromosome, you get all of those alleles together. Does that make sense? But what Morgan actually found is, so the way he set his experiment up is he would expect he would get a 1 to 1 to 1 to 1 ratio, but since they're linked, you would expect to get 2 of this kind and 2 of this kind. 2 of this parental that looks like this parent, and 2 looks like this parent, right? Because you're going to inherit your X from your mom and your X from your dad as units. You can only get those combinations. So you'd expect a 2 to 2 ratio or 1 to 1 ratio, right? And what Morgan actually got was essentially 1.9 to 1.9 to 0.1 to 0.1. These do not, this shouldn't exist. This is kind of like a mixing of stuff from mom and dad. And you can't do that because it's all inherited as one unit. And it was then that that people realized what these structures which they've been seeing in karyotypes might be. Remember the karyotype? I had that friend after college, she went out and did these karyotypes. It dropped the nuclei and the chromosomes would spread all out and then they'd line all the chromosomes up. Sometimes they would find chromosomes that looked really bizarre like this. And it occurred to them, maybe they're trading information here. And this is recombination in the process. So that if 2 X chromosomes line up next to each other, they might trade information somehow. So this crossing over is the explanation. So, the likelihood of 2 genes, 2 linked genes, 2 genes that are in the same chromosome, assorting independently depends upon the distance the genes are from each other. So, if we have this chromosome here, and we're interested in gene Y, gene W, and gene M. So that's yellow body, white eye, miniature wings. The odds of a crossover event happening here is much more likely that crossing over will happen here than in this smaller space here, right? And this was discovered very early on in Morgan's lab. The farther apart 2 genes appear to be on a chromosome, the more likely they are to recombine and assort independently, even though they're linked. Now, it's not absolute. There are hot spots. There are parts of the chromosomes that line up and just cross over really well. So it's possible to have 2 genes that are pretty close that assort independently pretty well. But in general, the farther apart, the more likely they are to get a recombination event in between. Questions on this? Okay, I'm going to skip over the chi-square analysis for right now. We may come back to that. We've already done that one. This is the one we're doing right now. Oh, that's with non-Mendelian, so we need 12. That's not right. I'm missing one in here. I'm missing a file. It wasn't on Blackboard either?
There was a spot for it, but there was no file.
There's no file. Ah, I forgot to upload the file with it. I'm in trouble.
The mapping.
Can you turn the camera off? This is going to take me a little bit to load this up. I really don't tell you Running? Okay. So, crossing over is a physical exchange of DNA, which we talked about before. And it happens, for the purposes of our class, it happens only during meiosis. If it happens other times, it's generally not a good situation. It would be a bad situation for the person. But the experiments, and I think it was Morgan then, it was the graduate students in Morgan's laboratory who actually figured out how to demonstrate crossing over. So they used what we call physical markers, and they used genetic markers. So a physical marker, what is a physical marker? So we're going to have this chromosome I'm going to draw here. And if we stain with this Giemsa stain, we get the banding pattern on here, right? So this one came from mom. This one came from dad, right? So a physical marker is an actual physical difference in the chromosomes that you would need to prepare a karyotype and drop the nuclei so they splat and spread the chromosomes out, and then you can physically look at the chromosomes. And in doing so many karyotypes in that laboratory, somebody found a chromosome on one of these flies. A piece of some other chromosome somehow broke off and got stuck on the end of this chromosome. So that one chromosome was larger than the other one. Even though they're homologous chromosomes, this has a bunch of extra DNA from a different chromosome sitting on it. Okay? And somebody also found a mutant form of that exact same chromosome in a different fly, probably years later or years earlier, whatever, years earlier, that was missing piece of DNA. Basically, some of the DNA had been damaged, and the damaged DNA had somehow been removed, and the chromosome had been closed up, such that this chromosome looked like this. And this band here is missing. Does that make sense? So if you do a karyotype and you stain with Giemsa stain, you do a karyotype, you can see that this guy is different here and this guy is different here. So that's a physical marker, or it's a genetic marker. Huh? Yeah, differences in sequence. Because they didn't have sequence there. How does differences in sequence manifest? You know what I mean, what I'm asking?
Yeah.
Different physical traits. Brown eyes, blue eyes, these types of things. Red eyes, white eyes, gray body, yellow body. You see the physical difference in the organism. It's called a genetic marker. You can see what the sequence is by looking at the organism. Okay? So, turns out this chromosome that had these weird things on it is the chromosome that had red eye or white eye gene on it, yellow body, gray body, and normal wild type wing or miniature wing. All three of those genes are on this chromosome. And for simplicity's sake, we can say, okay, well this guy here has red eye here, has gray body here, and has wild type wing here. And then in a different line, we can get this mutation where the white version of the eye color allele is here, the yellow body is here, and the miniature wing is here. So this organism is a carrier, right? It's dominant at these three and it's recessive at these three. If something inherited this and this, it would be white eyes, yellow body, miniature wings, right? So what they did is they got a pure breeding line of this and a pure breeding line of this, and they crossed them together to make these heterozygotes, the carriers. And then they did something that we call a test cross, and I haven't defined this test cross for you. We're going to do that now. So here's how it's represented in most textbooks. There's this piece of chromosome hanging off here, and then there's this deletion here. Looks like they used carnation-colored eyes instead of red and white eyes, so they used carnation eyes. Carnation means not quite as red as red. All right? So I actually give you a picture. Well, you guys can't see that. Sorry to you on the camera. I have to turn this up so people in the class can see it. This is white-eyed organism. This is wild type, brick red. Actually, that's probably apricot. Is a mutation in the eye color, and that might be carnation right there. And a different marker that they actually used was eye structure. So I want you to look at this. These guys have wild type eye shapes. They're described as almond shaped. They're actually mutations where the eyes appear as little bars. See how that's a reduced structure? Those are genetic markers. You look at the organism and you know what the genotype is, right? So I'm going to turn the lights on. Thank you. Appreciate that. So what they do with a test cross is get your pen ready. Write this down. A test cross, by definition, a test cross is always crossing a heterozygote, that is always crossing a carrier with something that is homozygous recessive. So the parental was pure this, crossed with pure this, right? I'll give you guys a second. By definition, a test cross is when you cross a heterozygote with a homozygous recessive. So when someone want to come up and do a monohybrid cross, which is a test cross for trait A, gene A, show the class what that definition is in a visual term. Come on up. What's your favorite color? Royal purple? Blue. Okay, there you go. Just like right here. Yeah. Right, that's a test cross. Yeah, you don't need to do it. Right? Right? When you have a heterozygote, a carrier, that you cross with a homozygous recessive. Thank you. So the reason I did that is not everybody is a text learner. Some people are visual learners, like me. I was driving around Houston and they wanted to give me directions. I looked at the map and said, stop. You know where I'm going. I looked at the map. Now I have this picture of wherever I go. You guys do that? No? No? You're not a visual learner?
I am just now with directions.
Ah. My wife, she prints out the map questions, directions, and she follows them exactly and reads where she's going. Just look at the map. Some people listen to the GPS. When I do that, I often will hear recalculating. Right? You guys know what I'm talking about? That's your little commercial break here. Okay, so what we've got, the parental population was this guy with this thing hanging off, pure breeding, and this one which is a little smaller because it's got the deletion, right? So what we've got here is the heterozygote. That's what we're displaying here. So the test cross of this is this guy. So we cross that. So this is no longer, so here we got the parental population. Here we have the F1, and we're not self-crossing. Up to this point, Every cross I've talked about, we've self-crossed these guys on themselves. Now to do what we're going to call genetic linkage mapping, we're going to cross the F1 in a test cross instead, okay? Very important point. What happens is genetic recombination can occur in here. Let me do this in a different color. So the reason we use something that is homozygous recessive is that the progeny organism will always inherit recessive, 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 right? Which means whatever gene is over here, whatever allele is over here, will definitely be seen and known just by looking at the organism, right? So let me do this. So this guy can inherit this, or could inherit this, right? Right? If crossing over never occurred, what does this organism look like? It's what? Dominant traits always, or? All recessive traits. Yeah, because these are all recessive. So we've got white eyes, yellow body, miniature wing, right? And this organism looks dominant at every gene, right? What if I do a karyotype and I have this karyotype and the organism is red eyes, yellow body, miniature wings. Crossing over occurred where? You want to come and tell me where? Where on here did crossing over have to occur? Between here, somewhere in here. Right? And since it's long, probably it happened like right in this area right here, right? Does that make sense? Okay, remember the videos are online if you want to run through this again. I'm happy to walk through this in my office. Yeah?
What does the actually DNA have to do with anything?
You can, when you do a karyotype and the nuclei splat out, you can look at the chromosome and say, this guy has that extra piece. I shouldn't have it on there, should I? Does that make sense? It shouldn't be on there, should it? Huh? Did I totally screw everybody up? So what? Okay, to make this, let's redo this. This is what it looks like. Okay, so it must have this. Right? So it's got red right here. But to get yellow onto this red, crossing over had to happen like right here, right? To move this guy over. Somewhere in here, crossing over had to happen, right? So now what we've got is we've got yellow on here. And then this appears to have gone over here also. And since this guy came over, this guy doesn't have that attached there. Does it make sense?
So if that mini didn't go over, it would have that extra thing on the bottom?
It was only yellow body change.
Yeah, if the yellow, if it just changed yellow and gray, nothing else changed.
Okay, so let's do that. This is great. So let me draw this out. I'm going to draw the chromosome And I'm even going to give you what it looks like, and I want you to tell me where the crossing over occurred. Okay? This thing is red eyes, yellow body, and miniature wings. It's not impossible.
Wouldn't it cross over just the same?
Can it cross over two places? Yeah. Do you see where it crosses over? Do you want to give it a shot? I'll just humiliate you if you're wrong. That's all right.
Yeah, we just did that, right?
Okay. You want to come up, Zach? You want to do it? Saved. You owe Zach a beer.
So there has to be an extra one here to get that tail.
Yep. Yeah, that's what happened.
Oh.
Got it? So crossing over occurred to bring these guys over onto this chromosome. So we've got red, yellow, miniature. But that means this thing is now over on this chromosome. The only way you could get this thing back is for another crossing over event to happen between those exact same two chromosomes during that one meiosis. Crossing over happened twice on this chromosome. And you can map where it happened by what you're looking at. How cool is that? That is like 1910, 1915, really cool stuff, isn't it? That gets you a PhD.
Okay. Sorry. So some guy just like found that and was like, "Ooh, this looks cool." Like,
Yeah.
That's super random.
Think of how many karyotypes they did before they were like, oh, this chromosome's totally screwed up. And you know, it's kind of the funny thing is, normally when people are like, oh, my pure breeding line isn't pure breeding anymore, there's this weird chromosome thing hanging off. Most people would be like, but as a geneticist, you have to say, oh, this thing's totally screwed up. Awesome! Now that it's totally screwed up, I can actually go against the normal system and I can learn about the normal system. So actually, there are people who will, we talked in lab, ethidium bromide causes cancer. There are people who will like grow their Drosophila in a culture of ethidium bromide because they want the ethidium bromide to damage the DNA and screw up the Drosophila because they want to spit out weird mutants. Because the weird mutants allow them now to do genetic tests back to see what the situation is. Scientists need to do the opposite. Oh, here's a virus, HIV, that's killing the human population. Awesome! Let's mutate it and do good things for ourselves. That's what we need to be doing in biotechnology. Things that are terrible for us are actually we need to use for good purposes. Okay. So, I think I've beat that to death. So, the graduate students from Thomas Morgan's laboratory said we can actually measure how frequently recombination occurs between two genes. So if recombination happens in here in, say, 5% of the population, in here it's going to happen, I don't know, 15%? And so we're going to call these, this is 5 map units of a chromosome. And this is 15 map units of a chromosome, or 15% recombination frequency that happens. And to honor their mentor, they decided that they would call a map unit is measured in centimorgans. I'm going to use map units, okay? So what they would do is they would do this, and they would run the test cross, and they would count 500,000 flies. And they'd say, out of 500,000 flies, I had 10,000 that were recombinants between these guys. That allows me 10,000 divided by 500,000, that tells me there's a 0.5 map unit space between here and here. Did I do that right in my head? Right? You can calculate the recombination frequency. So, they can run these kinds of experiments. So we have, this is Drosophila. This is actual data. from one of the chromosomes, and we can actually create maps of chromosomes saying where different genes are located in relation to each other on the chromosome. So, Y stands for yellow body. So I don't know, I've got a chromosome here like this. And I don't know where it is, so we're just going to say yellow is here. And the gene so that goes for yellow body. The gene for white eyes has a 1.1% recombination frequency. So it is 1.1 map units from the yellow gene.
How did you decide to go up or down?
I just randomly chose it.
Okay.
So this is 1.1. Okay. This is vermilion eye color. So the difference between yellow body and vermilion is 33 map units. So it could be 33 map units this way, or it could be 33 map units this way. Right? I don't know if vermilion is this way or if it's this way. Okay? So let's do vermilion to white. Let's run that cross.
Okay, thank you.
And we would expect if this is 32, then vermilion's down here. Or sorry, if it's 34, vermilion's down here. If it's 32, vermilion is up here. So white with vermilion is 32. So vermilion must be up here. Okay? And then we have yellow with miniature wings. So that's 34. So miniature wings is either here or way down here. So if miniature wings is up here, vermilion should be like one map unit apart. We do vermilion map, and it's actually 4 units apart. So this is 34. And this is what? 33. This needs to be 4. Why is it not 4? Just recombination does not happen randomly throughout. Relatively randomly, yes. There must be a recombination hotspot here. Or numbers, when you're counting this stuff out, they didn't have a large enough n, if you know what I'm talking about. So we can actually map all of this out and say where these guys are in relation to each other. But the maps are imprecise. If you're comparing to yellow, these guys are really close to each other. It turns out they're 4 apart. And when you get into where is the disease that causes this human ailment, you can't be like, it's sort of like on that quarter of the chromosome. Oh man, that's like 100 genes. You want to map things down very, very precisely. And then you can say, oh, there's 3 candidate genes here that could be the cause of this ailment in humans. So that's where we're going to get to a 3-point cross. In this case, they were comparing recombination between yellow and white. And then in a whole separate experiment, someone in a different part of the lab was doing yellow vermilion. And then a different person was doing yellow miniature. You put all the data together to do this. It turns out, if you do 3 genes simultaneously, you can nail it exactly. Okay? So you need to cross, just like I showed over there, yellow, white, vermilion. You follow all of that in one cross. It's a Dihybrid Cross. And you run it exactly like this. You have a pure breeding line, you breed them to get a heterozygote at each of the 3 genes, and you do a what cross? A test cross, a homozygous recessive N. So all 6 alleles, 3 different genes, 2 alleles at each gene, are all recessive. You cross that in, and you can get an exact map. You can get down to 1 or 2 genes. You can nail where in the chromosome it is. All right. I'm going to get started on this. I don't think I'm going to finish here. Maybe I'll wait.
You go to like kind of split.
That's a I can't do that very quickly. I can't cram this thing in at the end here. I know we've got 10 minutes left of class. I need you guys on Friday to have not partied on Thursday. I need you to have your brains, because I laid all this out as you and I have talked about it and we think about it. What happens is, you see how I've drawn all this stuff out? If I'm in a laboratory and I'm doing this all day, every day for 7 years, I'm not interested in writing little b, big B, little b, big P, little p. They do a shorthand. They figured out a way to illustrate all of this data with very few pencil marks on the paper. And anytime you would go into a real genetics lab, you have to understand the language they speak. So I need to teach you that language. I need to show you how this I totally understand. It's going to take me too long to explain this today and then run through the whole process. So we'll do that on Friday, okay? All right, cool, you're out of here early.